Hello, my name is Luana Mark, and I'll be presenting a paper to you written by Solange Duhamel and his colleagues by the name of Microbial Response to Increased Temperatures within a Lava-Induced Hydrothermal System in Iceland, an analogy for the habitability of volcanic terrains on Mars. In the paper, they studied a Mars analog on Earth to investigate how life could have formed under extreme circumstances, and further what to research on later missions. They did that by studying the eruption of the volcano Hularan in Iceland. It erupted 2014 to 2015. And what they did was study the environmental conditions, especially the thermal and physiochemical properties of the environments, and also what kind of microbial communities survived within this lava-induced hydrothermal system. Before we continue to the actual research, I would like to explain to you what actually a hydrothermal system is. It usually consists of a heat source, a permeable reservoir so the fluids can flow and trigger the active circulation, and a recharge system with, of course, an impermeable cover. They are formed through interactions of lava flow and near-surface water, or even melted ground ice below. Also on Mars, analogous hydrothermal systems have been discovered by the Spirit rover, which characterized the Joseph crater, the Jezero crater, and the Athabasca Valle's flood lava. And in the pictures what we see is first the crater here at the top, for instance the Joseph or Jezero crater, and then on the bottom we see how the lava induces a hydrothermal system, so with the ice beneath which gets melted and then starts circulating, and these explosive lava-water interactions. Why is it even important to study such hydrothermal systems? So first of all, and most importantly, hydrothermal systems are thought to be logical candidates for origin of life on Earth, but maybe even on Mars, as they provide a heat source and a protective shield from the extraterrestrial environment, so like a high radiation on Mars. And also they have the potential to preserve evidence of microbial life, also because of that shield. And maybe also they are able to remobilize dormant microorganisms through the heat source. So the lava here, on the right side we see the dormant microbes below the ground ice, which are then potentially remobilized by our heat source above. And it is also of great importance to astrobiology, because with these studies we can create or we can identify model organisms to then later inform in situ and remote sensing studies and guide current and future sample return investigations. So for instance, the European Space Agency's ExoMars and NASA Mars mission in 2020. Here we see a little picture of a rover at a hydrothermal system ready to sample. The method for mapping and the geologic context that they used was a unoccupied aircraft system, short UAS. It's a form of remote data sensing to create a digital terrain model and a stereo-derived orthomosaic, so like Google Earth, but way more precise. To measure the concentrations of nitrate, phosphate, sulfate, ammonia, sulfide, and chlorine, the DR900 multi-parameter handheld colorimeter was used. And to measure the chlorophyll A concentration, they used a acetone extraction with acidification method. And to measure the pheopigments, a Turner Trilogy laboratory fluorimeter was used. To measure the cell abundance, they first had to fix the sample at the sites in a 0.25% paraformaldehyde solution. Then they had to store it in nitrogen liquid until they could analyze it in the lab. And for the identification and enumeration of similar cell groups, they used flow cytometry. For the DNA extraction from the water samples, they first had to filter the water using a DNeasy Power Water kit. Then to purify the kit, Master Pure gram-positive DNA purification was used. For the analysis of the sequence, I used the 16S ribosomal RNA gene sequence analysis. And for the classification then of the closest known relatives, the software sequencer 5.2.4 was used. And further, for the identification of the isolates, they performed the BLAST searches in GeneBank. For the incubation, the water samples was put in bottles, and then for the different temperatures, they just put the bottles into different water sites. So one in a cold river, a glacial river with 5.7 degrees Celsius, and the other in a lava heated stream, which was 17.5 degrees Celsius. The main results for the mapping in geologic contexts was this map. So in A, we can see an orthomosaic with the sampling sites ranging from 1 to 8, and even the sample ID. So if we have sand, gravel, older lava. And then in B, we have the geological and the hydrological conditions. So now, additional to the sites, we also see if it was hot spring pools or a glacial meltwater stream or sand. So it gives us good information about the sites. The results for the water chemistry are very important for the study because it gives us information about the environment and also if the organisms could also survive or if the concentration of the chemicals are toxic. So the one thing that was really important to measure was the nitrogen and the phosphate because the ratio N to P provides a proxy for the nutrient stress. And here we see that in the water table and the pool. So in S, that's the site, and the pool, site 7, the ratio was below 16, which means that we have a biological growth limitation or a stress by nitrogen. And in the glacial river, so site 8 and the warm stream, site 4 and 5, the ratio was above 34, which again is a biological growth limitation, or it can also mean a stress by phosphor. For the sulfate, they couldn't detect anything, so 0 milligrams per liter. The following, so ammonia, sulfide, and chlorine, can be toxic over a certain threshold. And for ammonia and sulfide, they were both below the threshold in all the sites that they were detected. And the chlorine wasn't detected in the glacial river and the lava heated stream. So glacial river, again, we have here site 6, was non-detected. And here also non-detected in the warmer streams. But in the pool, site 7, they could find a 0.2 milligrams per milliliter. Further, we have the main results for the chlorophyll and pheopigments. They're both very important because they're easily detectable biosignatures by spectroscopic methods. And most of you might not know what pheopigments are. These are non-photosynthetic pigments, which are also a degradation product of algal chlorophyll. So, in the cool water, the concentration for chlorophyll A was very low, so from 0.02 micrograms per liter to 0.03 micrograms per liter, which indicates a limited plankton growth in the colder water. And then in the lava-heated stream, they found concentrations ranging from 0.06 to 0.07 micrograms per liter, which indicates a less limited plankton growth. Also, the degradation index, which is calculated through the concentration of chlorophyll divided by the sum of chlorophyll and the pheopigments. In the warmer streams, they got a 35% to 44% degradation index, and in the colder streams, a 28% to 12%, which indicates that there's a large photodetritus degradation, and the photodetritus abundance is relatively high. For the cell abundance, it was measured in concentrations times 10 to the power of 3. And in the background water, so site S, they found a 1.5 times 10 to the power of 3 cells per milliliter concentration. And in the lava heated stream, they found more, so 163 to 225 times 10 to the power of 3 cells per milliliter. We see that here, which means that in the lava heated stream we have a profitable cell growth. But then in the glacial river, so site 8, we only have 110 times 10 to the power of 3 cells per milliliter, which in return indicates that we have a less profitable cell growth. But then the cells were very high in the craters. So in the BT craters, we have 3,744 to 5,002 times 10 to the power of 3 cells per milliliter, which is 23 to 31 times higher than the lava water. So very profitable cell growth. Here, the following table gives us a very nice overview of the taxonomic composition of microbial communities. And we see that the chloroplast sequence are very abundant in sites S, so the background water, and the sites 2 and 3, which are lava heated streams. Also, the chloroplasts are an indicator for eukaryotic phytoplankton. But they didn't detect eukaryotes themselves, only the chloroplast. But this was expected since the primers they used targeted the 16S ribosomal RNA. In sites 4 and 5, the lava-heated streams, we get a large proportion of cyanobacteria. And in the glacial river, site 8, we don't get any chloroplasts. But then after 5 days of incubation, so C and W, we see that photosynthetic organisms were present, but they needed an increase in temperature to grow, which indicates that the organisms might have been in a state of dormancy, which have then been activated by the warmer treatment. This table is very similar to the one before, but here we see a comparison of the major bacteriophilia. So when they have a overalignment of more than 1%. And in the glacial river, we see that the sulfuricurvum of the Campylobacteriota was very present with 88%. And in the background water, we have the most abundant bacteria, the Geobacteria of the phylum Proteobacteria with 60%. And then in the lava heated sites, so sites low to 5, we get lower taxa, but we see that the cyanobacteria are most restricted to these sites. So here in green. From the incubation experiment, they could see how the microbial communities responded to the rapid temperature increase. In the beginning, they had about 90,000 cells per milliliter. And then after the 5 days warm treatment, the cell increased by a factor of 1.45, which is an addition of 40,700 cells in these 5 days. Then for the nitrate, they found a drop of 0.30 milligrams per milliliter. But then for all the other parameters, there was no significant change. So I have like 0.01 milligrams per milliliter. And also the nitrate drop by 0.3 could be a possible microbial growth limitation. So they don't have enough nitrate to take up. So the results they got are really cool and they actually speak for themselves. But it's important to always keep a critical perspective. So we used a volcano or a hydrothermal system on Earth as a Mars analog. So on Mars, the environment still is different. For instance, the gravitational acceleration or ambient temperature, pressure, atmospheric composition, the water availability, redox environment, organic availability, and the radiation. So many factors that we can predict a little bit, but not really, and it won't be exactly the same as on Earth. And also the way the heat is transferred, so the heat transfer rate will be different and could have an effect on any organisms, if there are any. And also, when we analyze the data sets, so when you analyze 16S ribosomal RNA gene data sets, you have to take great caution, as predicted by Bowman and his fellows. And also the microorganisms in the water samples could have been transported by bioaerosols, which is also presented in the paper by Perfumo A. So it could be that they originated in the hydrothermal system. It doesn't have to be. For the conclusion, I present you here a short summary of the main results. So, in the lava heated stream, we saw that there was an increase in the microbial abundance, the richness of the microbes, and their diversity. And also the metabolism shifted from lithotrophy to organotrophy. We had lithotrophy in the glacial river, which was dominated by the sulfuricuvum and the organotrophic bacteria. And then phototrophy in the warm water, where the cyanobacteria dominated and the macroalgae. And also, new taxonomic and metabolic groups were identified with the sequence similarities to subsurface ecosystems. For instance, the thermophile candidatus acetothermum autotrophicum and the endospore former Geobacillus stereothermophilus. Overall, I think these studies are really great and they give us an important understanding for the microorganisms in a lava-induced hydrothermal systems, and also a great understanding of the geochemical environment associated with such a hydrothermal system. Also, we can determine whether these studies are applicable to Mars, and by taking a Mars analog on Earth, firstly, it's a great possibility or opportunity to study Mars because it's here on Earth and also it's way cheaper. Then for the flaws in the methodology, so some concentrations of the chemicals were very small, which are also hard to detect. And also with the DNA extraction, there was a lot pipetting included. There you also get more mistakes when you have to pipette a lot because sometimes it gets stuck in the pipette tip or you don't get everything up. So humans make mistakes. Then for the outlook, I think it would be really great to apply the same methods used in this study for other volcanic eruptions on Earth. By asking the questions, can similar lava-water interactions generate habitable environments? So when we not only have this one volcano studied, and to get a broader understanding which microorganisms are formed in such hydrothermal environments. And also with such studies, we can work together with future Mars sample missions. And what would be really cool to discover, obviously, are dormant microorganisms on Mars. Like here we see sleeping microorganisms, which are woken up by the volcano. Now I'm more than happy to answer any kind of questions you may have.